Hey everyone, my name is Chris Anderson. Our bodies are made of cells, but did you know that many of those cells are symbiotic bacteria? Millions of bacteria live inside of us, helping us survive. In fact, most of you isn't even you. Dr. Senu Apewokin is a researcher at UC Health, and he and his lab are learning more about the microorganisms we live with. Hey Senu.
Hi, Chris.
Thanks for inviting me down to your lab, man.
Thank you. You're welcome. Always good to see you.
It's good to see you too. So, what is a microbiome? What is our microbiome?
So microbiome is a term that we use to refer to the collection of all the bacteria and the genes and everything that lives in the environment, in that environment. So we look for an environment and all the collection of genes. They can come from bacteria, they can come from the host cells, like for the human beings, it can be the human cells. All that collection is referred to as the microbiome.
So when you say environment, you mean like wherever we're talking about inside our bodies?
Could be inside, it could be outside. So we can have the skin microbiome.
Oh,
so
wow.
when it's on the skin, we call it, that's the microbiome of the skin. We could have the most common people hear about is the gut, and that's called the gut microbiome.
So what do the bacteria that live in our gut, why are they there?
Those bacteria also help us break down the food we eat, and they are able to break down the food into components that the body can use to make energy, to grow cells, to grow muscles. That's what the bacteria help us do. But they also influence our health in other ways, like they control our ability to respond to infections and things like that. That's also controlled by the microbiome.
So the bacteria inside of our gut, they help us break down food, which is probably really great. But they also help us keep other diseases out?
Correct. So there's always a battle between good bacteria and bad bacteria going on all the time. And when the good bacteria wins, the body's happy. Sometimes when the bad bacteria wins, it causes us to be sick. And some of the bad bacteria, sometimes when the bad bacteria wins, diseases like C. difficile can be present. So C. difficile is one of those organisms that manifests or shows itself when bad bacteria are winning the battle within our guts. So that's what I study.
So how do you simulate the gut out in the lab instead of actually going into a person and making all your experiments?
Very good question. So to do that, we try to get cells from human beings and grow them in the lab. So
you're growing human cells, human gut cells?
Correct. Human gut cells in the lab.
That's cool. Can you show me how you do that?
Yes.
All right.
I'll show you.
I'll follow you then.
Okay. So what I have here is just a microscope, and with that microscope I can look at the cells.
Okay.
So what I'm going to do is take some cells out of the incubator.
That's the incubator right there?
Right.
Okay.
So this is an incubator, and what it does is that it keeps the cells around 37 degrees. That's our body temperature.
Okay.
And
37 degrees Celsius, right?
Correct.
Got it.
Correct. I'll pick up the cells and then I'll show you, what they look like when I pick them out. So I'm going to show you two groups of cells. The immortalized cells, they're called Keiko2 cells.
Uh-huh.
These cells have been growing for a while, so you can see here, and I'm going to explain.
So the cells are living in this liquid?
Yes, they're living, so they're coating the plate at the flask.
Okay.
And the liquid is on top.
Oh, so they're growing on the bottom there.
Correct.
Okay.
So they're growing like a carpet on the bottom of that.
Okay.
And the liquid provides the nutrients for these cells.
All right, so they're on the bottom, they're carbonated. So we're going to take a look under the microscope.
Correct.
All right, let me take a look. Oh, so those are all the cells inside our gut?
These are cells inside our gut.
Yeah, they are. They're just kind of everywhere.
Right.
Where would I find these cells in my body? The gut's a pretty big place. Where would I find them?
So these are epithelial cells.
Epithelial cells.
Right. So epithelial cells are, pretty much the first line of defense between the bacteria in your gut and then within your
Wow, that's cool. So these are immortalized cells that you can run different trials on then.
Correct. We also have cells that we've grown from stem cells. And stem cells are those cells that have the ability, depending on the signals they get, to grow into an arm or a leg or
your lungs or your ears or...
Correct.
Okay.
And in this case, what we do is we take these stem cells and grow them into a small intestine.
So when you're doing these experiments, you've cultured these cells, and then you can expose them to different bacteria that you want to see how those cells would react, whether they're healthy, or you can put in medicine. That's how you run your tests?
That's correct.
That's really cool.
That's correct. That's exactly how it happens. So this is a transwell system.
That's a little well right there?
Correct.
Okay.
So this is a plate that has these baskets in there, these small baskets. So the baskets are within, each has a well, and then the baskets are within the well. So we call them, this is a well, and this is the basket.
That's the basket, okay.
Right. At the bottom here is this membrane, and the membrane has holes in them.
Okay, how big are the holes?
They're very, in this case, they are 0.4 microns.
So they're really microscopic holes.
Yes, microscopic. Yes, microscopic.
So what sort of things do you put either in the well or in the basket?
So different things. So a lot of times we put drugs in the well.
Drugs in the well.
So we use sometimes in my lab, or we study chemotherapy effects on cells. So we put chemotherapy in the well. And then sometimes we put bacteria in the basket.
Okay.
So we can put some C. difficile in the basket. We can sometimes even try to put stool in the basket. So we can really tell if there's a leak, then the cells are dead or destroyed, and that's why there's a leak from the top to the bottom.
And if it doesn't leak, then you know whatever either chemotherapy or medicine is then working to keep those cells healthy.
Correct.
That's very clever. So how can you tell if there's a leak?
There are several ways to do that. You can put a dye on the top well.
Okay.
And then look for the, and if it leaks into the bottom well, you look for the dye in the bit. That's one way to do that. Another way is we measure electrical resistance. So we put an electrode on the top well.
Okay.
And I put another on the top basket, and I put another in the bottom well, and look and see if the current can get across from the top to the bottom.
Oh, and if there's no current, then those cells are healthy, they fought off that other bacteria, the medicine's working.
Correct.
Well, Senu, thank you so much for inviting me down to your lab and teaching me all about like the bacteria and the things that aren't even me inside my body. That's really cool.
You're always welcome, Chris.
Thanks. I appreciate it.
You're welcome.
And thank you for watching. We'll see you next time on Science Around Cincy.
Intro, take one.
They have fun. They have fun in this project. All right.